This is part 2 of the statistical analysis of quantitative traits. And I want us to start with what we call the various components of quantitative traits. So as we discussed earlier, the metric value or the phenotypic value for a specific individual results from genetic factors, environmental factors, and also environmental factors that interact with genetic factors. So usually we can sum these factors in a population of individuals, segregating for a quantitative trait that contributes to the variance of that population. So the total variance in this case, that is normally indicated as VP for the population, is therefore as a result of variations in genotype or genetic factors, environmental factors, and genetic factors interacting with environmental factors. So the VP is the one we are saying is now the total phenotypic variation of a segregating population for a particular quantitative trait, and it is given by genetic variation, which is VG, that contributes to the total phenotypic variation. Then we have VE, the environmental contribution to the total phenotypic variation. And now the VGE, which is the variation associated with genetic and an environmental factor interactions. Yes? So the genetic variation can then further be divided into 3 components. And we mentioned this, that the first component is the additive gene factors, which we call it additive genetic variation. And some alleles may contribute to a fixed value, may contribute a fixed value to the metric value of the quantitative trait. For example, if genes A and B control the corn yield, it is actually controlled by many genes, and each allele is going to contribute differently to this yield. And this is now like what we had illustrated earlier, that maybe the dominant A allele will contribute 4 units, the recessive B allele will contribute, A allele will contribute 2, B 6, then the recessive 3, and so on. So if you have this genotype of dominant forms, then you will have a yield of 20, which is 4 plus 4, 6 plus 6. And if you have this heterozygous genotype, then you will have a yield of 15, which is 4 plus 2 plus 6 plus 3. I hope you remember these ones. So genes that act in this manner are additive because every allele is contributing to the genotype. So then we say that they contribute to the additive genetic variance, which is denoted as VA. So remember now we are looking at this first part, genetic variation, and we are giving it other factors that affect genetic variation. One of them is now what we have said, the additive genetic variance. In addition to the genes which have an additive effect on a quantitative trait, other genes may exhibit what we said, the dominance. So we have dominant gene action, which will mask the contribution of the recessive alleles of that particular locus. And that is what we normally say, for example, if the 2 genes just mentioned exhibited dominance, the metric value for the recessive heterozygote would be B, would be 20, because the 2 recessive forms in this case are masked and they do not contribute to anything. So being 20, it then resembles the completely dominant gene action. And that means that both the heterozygote form and the homozygous dominant will then give us the same phenotypic value of 20. So this value equals to the homozygous dominant genotype in the example where the alleles are acting additively. This kind of variability is therefore attributed to dominance genetic variance. So now we have another one that is genetic, which is dominant genetic variance, denoted as VD. And then we have another one, which is the final type of genetic variance in association with interactions between genes. So the genetic basis of this variance is called epistasis, and it is called the interaction genetic variance, VI. Usually here is where the one gene can affect the expression of another gene. Yeah. So there is gene interaction giving us a specific phenotypic characteristic. So then the total genetic variance that we have is therefore an addition of the three forms of variance. So VG is therefore attributed to the additive variance, we have the dominant variance, and we have the interaction variance. So these three contribute to our total genetic variance, and therefore they contribute to the total phenotypic variance that we had when we were beginning, and therefore we can rewrite this as the total phenotypic variance equals to, now instead of VG we have VA, we have VD, we have V1, and then of course the variance related to environment and variance related to genetic and environmental interaction. So by performing specific experiments, quantitative geneticists can be able to estimate the proportion of the total variance that is attributable to the total genetic variance, and also that is attributable to the environmental genetic variance. So if geneticists are trying to improve a specific quantitative trait, maybe they want to improve crop yield that we want to do, maybe, or weight gain in an animal, so that you can have more quality and heavy weight. So if we want to improve, then estimates of the proportion of these variances to the total variance provide the direction to this particular research. So for example, if a large portion of the variance is genetic, then gains can be made from selecting individuals with the metric value you wish to obtain. So if you find that from this equation that the greatest proportion contributing to the total phenotypic value is actually as a result of genetic characteristics, then you do what we call selection of individuals, where you breed the best of the best. And I think this has also been applied traditionally. So you look for the best animals if you want to increase meat in cows. So you look for the best and mate the best animals. Yeah, that way you increase the chances of having an offspring that has caught that kind of genetic gain. On the other hand, if genetic variance is low, if the phenotypic characteristics of a certain, let's say the same animal, a cow, if the weight is not actually attributed to genetics as much as it is attributed to environmental factors, this implies that then more success for you to be able to get the best animal with the most weight would mean you check which are the best environmental conditions under which this individual can be grown, so that you optimize that. Look at what is the environment. Do I need to give this cow specific food? Do I need to give it a specific shade? Do I need, all those, because those are now environmental factors. So you can see that this equation is very important. It helps us to know if this is the one that is giving us the highest contribution to the total phenotypic value, then I can either increase this or decrease this to achieve what it is that I want with any biotechnological gains or advancements. Okay. Thank you for that. Now we go to the last part of this statistical analysis.